Okay, today's lecture is on stem cell therapy and overview. So, we'll be talking today about stem cells, we'll be talking about the sources of stem cells, we'll be talking a little bit about the controversy surrounding stem cells, and we'll be looking at the current usage of stem cells in therapeutics. And then we are going to look at what's the future potential of stem cells in therapeutics. So, to understand stem cells, we need to look at development. Development in this context, we mean the steps involved in the processes from ovulation to implantation. Implantation where? In the uterus. Okay. So, what is ovulation? Ovulation refers to the release of the secondary oocyte from the Graafian follicle on the surface of the ovary. Okay, that is ovulation. So let's look at the diagrammatic representation. This is totally diagrammatic. So I'm trying to depict here the uterus, and somewhere here you'd find the ovary. Okay, so because it's bilaterally symmetrical, you can have an ovary there, and the ovary releases the ova. That ova is taken up by the fimbriae, it reaches somewhere there, and then we have the sperm coming up the uterus, reaching there, getting into the ova, and then the nucleus of the two cells fuse and we get what is called the zygote. The zygote moves down the fallopian tube. In the process, it's developing till it finally implants itself at the walls of the uterus. So that's what we're talking about here. We're talking about the steps from ovulation to implantation. Okay, so we are going to follow the path taken by the zygote till its implantation. So we are going to talk about this story from ovulation to implantation. Okay. So first event is ovulation. What's ovulation? The release of the secondary oocyte. So let's look at what's an oocyte. Okay. Okay, so this is a diagrammatic representation of an oocyte. Okay, this is the representation of an oocyte. The outer layer, it's the zona pellucida. Outside this is a layer that we refer to as the corona radiata. Okay, and this is the membrane of the egg. And somewhere here is the nucleus. This is the pronucleus. Of the egg. Okay. So now let's represent sperm here. So it's a diagrammatic representation of a sperm. Okay. So let's pick up the story again. The sperm comes up the uterus, goes into the fallopian tube, and somewhere there it meets the egg cell, which is released from the ovary, and then there is a fusion of the egg cell with the sperm. The sperm releases its nucleus into the cytoplasm of the egg, and then these two pronuclei, they fuse together to form the zygote. Okay, so that's what we are going to depict in the diagram here. So these two come together. Let's draw the egg again. Okay, and let's depict the pronuclei of the egg, and let's denote the pronuclei of the sperm. Okay, so that's the egg pronuclei. This is the sperm pronuclei. The next event is they fuse. The pronuclei and the egg pronuclei, they will fuse. That's the next event. Okay, so let's try to depict it again below. So this fuse to form a single nuclei. This we call the zygote. Okay, this again we are trying to depict the zona pellucida. Then we told that this zygote moves towards the walls of the uterus. During this time, it undergoes certain kinds of development. So that development is what we are now going to talk about. Okay. So what happens? Now, these cells will divide into the single cell, this whole single cell divides into two cells. At this point in time, the zona pellucida is a bit rigid. As a result, what happens is the cells divide without growing. Okay, so it doesn't increase much in size, but it divides. So this is the two cell stage. From the two cell stage, it becomes a four cell stage. Okay, four cell stage. From the four cell stage, it becomes a eight cell stage. From the eight cell stage, it becomes a 16 cell stage. So it becomes a 16 cell stage. This 16 cell stage morphologically looks like a mulberry. Okay. So this is called a morula. Okay, so morula means mulberry. Okay. From this stage, some changes take place. Okay. Some cells migrate to the periphery. Let's try to draw the cells in the periphery. So some cells migrate to the periphery. Okay. And some cells get compacted inside. Okay, so initially these cells are of similar morphology, but after the 16 cell stage, let's call this the 58 cell stage. Okay, so the number of cells might not be 58, but I'm trying to depict this as a 58 cell stage, and this stage again becomes a similar, this is 107 cell stage. Okay, so this stage we call it the blastocyst. Let's call this early blastocyst. This we can call late blastocyst. Okay, let's go back to the diagram and orient ourselves. So this zygote comes down, it becomes a ball of cells, 16 cells, we call it the morula. The morula begins to come down, and at that same time it is the cells within the morula are dividing. It's becoming from 16, it's going to 58, and later on 107 cells, at which time it gets implanted. So the blastocyst stage is when it actually gets implanted. Late blastocyst stage, we can say it gets implanted. Within the walls of the uterus. Okay. Okay, the important thing to remember here is that these cells up to the 16 cell stage, so let's write here up to the 16 cell stage, okay, they are having similar morphology and they are more or less always similar to each other. So if we take a cell from, say, here, bring it outside, this cell has got the potential to become a whole organism. Okay, so up to the 16 cell stage, each cell has got the potential to become the whole organism. So these cells we call totipotent cells. Totipotent cells. Okay. By the blastocyst stage, we said that some cells go to the periphery and some go inside. The one at the periphery, we call them trophoblast. The one inside, we call it the inner cell mass. Okay. The trophoblast cells later on become the placenta. The inner cell mass gives rise to the 3 germ layers. Okay. So the point is, this inner cell mass becomes the embryo. Okay. The embryo has got 3 germ layers. What are the germ layers? What are the germ layers? The germ layers are ectoderm, mesoderm and endoderm. Okay. The ectoderm forms the epidermal tissues and the nervous system. Okay. The mesoderm gives rise to the muscle, bone, blood, and the urogenital system. The endoderm gives rise to the interior stomach lining, the GIT, and the lungs. Okay, but it cannot form the placenta. Okay, it cannot form the trophoblast cells which later become the placenta. Okay. So, because this cannot form the placenta but can form ectoderm, mesoderm, and endoderm derived cells, we call the inner cell mass, the cells of the inner cell mass, we call them pluripotent cells. Okay. So, totipotent cells and pluripotent cells are stem cells. Potent and pluripotent cells are all stem cells. Okay. So totipotent and pluripotent cells are stem cells. Let's define stem cell then. Okay, so stem cells are cells derived from multicellular organisms, and they have got two important properties. One is self-renewal, and secondly, they can get committed and then they can differentiate. Okay, so they've got two important properties. What is self-renewal? One stem cell can divide and give rise to another stem cell. What is committed? This stem cell can become a ectodermal cell, it can become a mesodermal cell, and or it can become a endodermal cell. Okay, So for example, if it becomes a mesodermal cell, then it can terminally differentiate into a muscle cell, a bone cell, a blood cell, or a cell of the urogenital system. Let's take an example and say it becomes a bone cell. Okay, so stem cells have got two properties. They can divide to themselves, or they can choose a particular fate. They can become an ectodermal cell, a mesodermal cell, or an endodermal cell. So example, they can become a bone cell. So this bone cell is entirely different from the stem cell, and it cannot physiologically go back to becoming a stem cell. So a bone cell will divide to form a bone cell only. Okay, it cannot give rise to a stem cell. Such a cell we call terminally differentiated Okay, so a bone cell is a terminally differentiated cell. Okay. In addition, stem cells have got a property. They don't obey the Hayflick limit. Okay, so what is Hayflick limit? Says somatic cells. What are somatic cells? Somatic cells are the cells of the body except the gonads. Okay, so somatic cells can undergo division only about 35 times. Okay, after this the cells become senescent, meaning they become old and they cannot divide anymore. Okay, but stem cells don't follow the Hayflick limit. So the question is, why do somatic stem cells become senescent after 35 divisions? It is because they have got telomeres. Okay, so telomeres are ends of chromosomes. So these telomeres, with every division, they reduce in size. By 35 times, they are completely lost. And if telomeres are lost, a cell cannot divide. Okay, but we said stem cells don't follow the Hayflick limit. Okay, but they also divide. Okay, they also divide. So theoretically they should also undergo division only about 35 times, but no. Okay, they don't obey the Hayflick limit. They are kind of an immortal cells. Okay, they can continue to divide. Why? Because they have an enzyme called telomerase. Okay, so if you got telomerase enzyme, as the telomere shortens during each division, the telomeres will synthesis and maintain the length of the telomere. Okay, so these are the description about stem cells. So we have defined the stem cell and we have talked about the Hayflick limit. Okay, so let's come back to our slide and see where we are. Okay, so we have talked about the stem cells. Okay, we have tried to understand stem cells from development. Okay, and we have said there are two types of stem cells we have discussed in our discussion. One is the inner cell mass, and we have talked about the pluripotential stem cells. Okay, pluripotential. Um, did I get it wrong? Yes, I got it wrong. Okay, so we talked about two cells. Which were the two cells we talked about? We talked about totipotent cells and we talked about the inner cell mass cells. Okay, inner cell mass cells, and we said that inner cell mass cells are pluripotent. Okay, then what did we do? We looked at the definition. We discussed what a stem cell was, and we told that we discussed that it's got two important properties. One is self-renewal. What's the other property? It can get committed and then it can differentiate. The other thing we said that stem cells are immortal cells. They have got telomerase activity. Okay. So it's got it has got telomerase activity. Okay. So there is a question. Okay, which are the other cells? Other cells have telomerase enzyme. Okay. So if you have answered cancer cells, you are correct. Okay. Bacteria can grow indefinitely. Definitely. Okay. Because why? They don't have telomere. Okay. Telomeres are absent in the bacterial chromosome. Okay. So they can grow indefinitely. And so that was a trivia. Okay, let's now talk about the sources of stem cells. What are the sources of stem cells? Okay, for this we need to draw the blastocyst again. Okay, so we said that it has got a differentiated outer layer of cells. What was that outer layer called? Yes, indeed, trophoblast. And it had an inner cell mass. Okay, this is supposed to look the same size. Okay, so that is the inner cell mass. Okay, so our question is, what are the sources of stem cells? So we have told that the inner cell mass are pluripotential, and that they are stem cells. So, is this a source of stem cells? Yes. Okay, so if we can remove a cell from here, bring it outside. Okay, and that is a stem cell. Okay, that is a stem cell. So these cells which are derived from the embryo, they are called embryonic stem cells. Okay, they are called embryonic stem cells. Okay, usually they are derived from inner cell mass. Okay, derived from the inner cell mass. Okay, they are derived from the inner cell mass. The second source. Let's go back to the diagram. So, we said that the inner cell mass gives rise to the three germ layers, the ectoderm, mesoderm and endoderm, and they form the terminally differentiated cells like the muscle, bone, etc. Okay. Okay, they become the muscle, bone, etc. Okay. Now, the bone is a terminally differentiated cell, but within the bone there are some stem cells. Okay, within terminally differentiated tissues there are stem cells. Okay, so not only does the bone contain terminally differentiated cells, it also has some number of stem cells. Okay, so these obviously are found in the organism. It's not found in the embryo. Okay, so bone cell is not technically found in the blastocyst. Okay, only when it becomes a fully grown embryo, then you can see bone cells. But at this stage, in this diagram, we don't have bone cells. Okay, so in that aspect, we say that the bone cell is adult. Okay, so we can come down here and say that the other source of stem cells is adult stem cells. Okay. The third source of stem cells is from the cord cell. Cord cell and the amniotic fluid. Okay, so as the embryo continues to develop, a small cavity develops somewhere here. Okay, and that cavity gets filled with amniotic fluid. Okay, so this amniotic fluid is also a source of stem cells. Okay, so when the fetus comes out, okay, when the baby is born, what happens? The baby is attached to the placenta, so the trophoblast cells give rise to placenta. So the infant is connected to the placenta by the cord. Okay. Okay. So the cord. This cord contains blood. and those blood contains stem cells. Okay, so the umbilical cord contains blood, and that blood contains stem cells. Okay, so these type of cells, the stem cells which are present in the cord or the amniotic fluid, we give a special term to that. We call it perinatal stem cells. We have the perinatal stem cells. In 2012, two scientists, one from Japan called Shinya Yamanaka and John Gordon discovered something phenomenal. They took an adult cell, let's say a muscle cell, from an adult, okay, so these cells contain the genome, the genetic material, and they expressed certain proteins. There were 4 proteins: OCT4, SOX2, and c-MYC. Okay, they expressed these proteins. These proteins are nothing but transcription factors. So this resulted in this cell becoming genetically reprogrammed. Meaning they turned into stem cells, pluripotential. Okay, so the adult cell became a pluripotent stem cell by genetic reprogramming, and these were termed induced pluripotential stem cells. Induced pluripotential stem cells. Okay, so that is a source of stem cells. You can take an adult stem cell, you do a bit of genetic reprogramming, and you end up with a pluripotential stem cell, which we call induced pluripotential stem cells. Okay, so these are the 4 sources of stem cells. Okay, so what are the 4 sources? Embryonic stem cells, adult stem cells, cord cell, and amniotic fluid, which we call the perinatal stem cells. Okay, and the induced pluripotential stem cells. So there are 4 sources of stem cells. Okay, so let's go back to our slide and see where we are. Okay, so we have talked about stem cells. We have talked about the different sources of stem cells. Which are the four sources that we talked about? Embryonic stem cells. Okay. Then adult stem cells. The one that we get from the cord, what do we call it? What do we call? Perinatal stem cells. And the Nobel Prize winners discovered what? Induced pluripotential stem cells. Okay. So we have talked about all this. Okay, these sources of stem cells. Now we are going to discuss the controversies. Okay, let's go back to our diagram there. Okay, so here we said so we said that we can source the stem cell from the embryo. So we remove this cell, it becomes a stem cell. But in the process, what will happen? In the process, this embryo will die. Okay, we will get the stem cells, but the embryo will die. Okay, so we have to remember this embryo is a potential human being. Okay, if it is allowed to go to term, then it will become a human being. Okay, but here, because we are removing the stem cells, the embryo will die. So this is not we don't have ethical clearance. Okay, so there are ethical issues. Okay, so this is not a very good option. Okay, let's look at the second option, adult stem cells. Okay, so let's go back to our diagram. So in the beginning, there was a sperm and an egg. They fused together. They formed the zygote. The zygote underwent all these things. Okay, and then it became a blastocyst. From the blastocyst, what happened? The blastocyst gave rise to the 3 germ layers, and we got the terminally differentiated bone cell as an example. Okay, so now it has taken us time. Okay, so let's explain that with the help of a diagram here. So let's say first it was the zygote. Okay, so a zygote is typically formed about 12 to 24 hours after fertilization. Okay, this zygote becomes an adult, let us say 19 years. Okay, so T equal to 1 day and T equal to 19 years. From this 19 years adult, we are taking a cell and converting it into an induced pluripotential stem cell. Okay, so the cell had begun its journey from a zygote to an adult, 19 years, and that 19-year-old cell, we are converting it into a stem cell. So during all these years of life, about 19 years, what's happening? This adult cell which we selected is undergoing mutations. Okay, so these mutations may be lethal, they may be non-lethal, but such a mutated cell only, we are now making it a induced pluripotential stem cell. And if we use it for any other purpose, what's going to happen? This stem cell is going to carry the mutations. Okay, so that is also a problem with adult stem cells. Okay. So that is also the problem with induced pluripotential stem cells. Okay, so adult stem cells, we know that it is a stem cell which is present during the blastocyst, and it is now available in the adult stem cell. Okay, that is an adult in the adult cell. So that is an adult stem cell, and the other one is induced pluripotential stem cell. Both have got the same problem. Okay, because adult stem cells may also have undergone mutation. Induced pluripotential cell also may have undergone mutation. So that is also a problem. So then, which is the best source? The best source would be a cord cell derived perinatal stem cell. It doesn't have these problems. Okay, so what's the advantage of a perinatal stem cell? So the advantage is ethical issues are less. Okay, and mutations would be less. One cross this out, we can say it has got reduced mutations. But what's the problem? Suppose I want perinatal stem cell, but what's the problem? My cord cell has not been stored. Okay, so it is not an option for me. Okay, it's only an option for people who have stored their cord cell. So that is why you must have heard about cord blood banking. Cord blood banking. What is cord blood banking? So potential parents are told by these companies which do this to sign up, and if they sign up, these companies will come and collect and store the cord blood in their blood bank in a viable form, so that whenever the patient or the person needs the cord blood, they can get it from the bank, and that can become a source of perinatal stem cells. Okay, so these are the controversies and issues surrounding stem cells. Okay, let's go back to our slide. So we have covered stem cells, we have covered the sources of stem cells, we have talked a little bit about the controversies, and we have concluded that the cord blood cell is relatively a good source. Okay, there is one more thing that people have done. Okay, so to overcome the ethical issues, they did one more thing. Okay, one more thing. You must have heard about doling. Okay, if you have jumped up in your seat and said that it was the first cloned sheep, then yay, you're absolutely right. It was the first cloned sheep. So how did they clone? Okay, so we're going to learn the steps of cloning. Okay, so let's say cloning. What's the first step that they did? They took an egg cell. This egg cell has got a nucleus. Okay, they took an egg cell. Then they took an adult cell. This adult cell has also got a nucleus. Okay, so this is the nucleus. Okay, so they took, what did they take? They took an egg cell from the sheep. They also took an adult cell. What's the second step that they did? They removed the nucleus. What did they do? Removed the nucleus from the egg. Okay, so what do they get? They would get a cell without a nucleus. Okay, in the second stage, what did they do? They repeated the process with the adult cell. And what did they get? They got again an adult cell. The nucleus is out of the adult cell. Okay. Then they did the third thing. What is the third thing that they did? They took the nucleus of the adult cell and inserted it here into the empty egg cell. Okay, so what did they do? They took this and inserted into. Okay, so after that, what do you get? You get a cell, okay, with a nucleus. And this nucleus came from the adult cell. Adult cell, nucleus, and this is the egg cell without nucleus. Okay. Then they did a fourth thing. What's the fourth thing they did? Okay, fourth thing was they gave this cell an electric shock. Okay, then what happened? This cell became an embryo. Okay, they took this embryo and put it into a female sheep, and what happened? Dolly was born. Okay, so if you have an embryo, definitely you have got stem cells there. So this could be a source of stem cells, but again the problem is embryo. Okay, ethically this cannot be a source of stem cells, because the same process can be done in human beings. Has it been done up till now? No. Okay, can it theoretically be done? Yes. Okay, why haven't they done it in humans? Ethically, we don't get permission to make a clone of a human being. Okay, so what is the difference between this embryo and the physiological normal embryo? So normal embryo, it needs a sperm and it needs an egg. Here you need an egg, okay, but you don't need a sperm. Okay, you need only a somatic cell nucleus. Okay, but both of this can produce an organism. Okay, in this case we call it a child. When we do this, we call it a clone. Okay, so this is the step of cloning. So cloning can also be a source of stem cells. Again, the issue is ethical. Okay. Okay, let's see where we are in the contents. we have completed the controversies regarding stem cells. Okay, so now we are coming to this topic. What's this topic? The current usage of stem cells in therapeutics. Okay, so the current usage, which is very successful, is bone marrow transplant. Okay, what is BMT? Bone marrow transplant. Okay, it's done for what patients? Usually cancer patients. Okay, so what is the idea? The bone marrow, 1 in 15,000 cells of bone marrow is a stem cell. So the idea is, if you transplant bone marrow, 1 in 15,000 cells is a stem cell. This cell can form the granulocytes, monocytes, agranulocytes, let's rewrite it. Form the granulocyte, agranulocyte, and platelets. Okay, so this is the idea why we are doing bone marrow transplant. Okay, but the efficiency, you see, it's 1 in 15,000 cells. Okay, so it takes time for the stem cells to be established. Okay, and these cells to be formed. So how can we make the bone marrow transplant better? If we can separate the stem cells and concentrate it, and then if we transplant, that would be a better way to do it. Okay, so lots of bone marrow transplants have been done. They have been found to be very successful and effective. So there are other uses of stem cells in therapeutics, but the number of patients is less, and therefore I have not included it as part of my presentation, but you can always look it up. Okay, the other uses of stem cells in therapeutics. Okay, so what is the potential? Okay, the potential is, let me illustrate it with an example. Suppose there is a person who has a pacemaker. Okay, so a pacemaker is a prosthetic device, meaning it is programmed outside the body to generate impulses. Okay, so it cannot respond to local changes in the heart. Okay, so this is the problem with pacemakers. Then again, the body can reject it if it's immunogenic. Okay, and then it is an external thing which has got a battery. The battery may fail. Okay, so there are lots of issues with prosthetic devices. So let's give another example. A person has got, say, maybe diabetes, say type 1. Okay, so type 1 diabetes is basically you don't have a functioning pancreas. So what if you can make a pancreas using stem cells from the patient himself? So what will happen? It won't be rejected by his body, and this is physiologic. Okay, so he wouldn't have to potentially use insulin all his life. Okay, his body will become normal. and it can change a person's life. So this area of research is called regenerative medicine. Okay, medicine. Okay, so where you can grow spare parts for the body. Someone has lost its leg and has a prosthesis. So you can you can make a new leg. So that is the potential. Okay, it's potential. Okay, and some of it is experimental. Okay, so but this is a life-changing area where stem cells can contribute. Okay, and it can change the life of so many people. Okay. On that positive note about stem cells, let us stop our lecture here.